Okay, so for the last installation of linkage, meiosis and the single gene, we're actually going to do 3 genes, 2 linked, 1 unlinked. Okay, and we're going to move away from phenotypes for now. We're just going to sort of speak abstractly, and we're going to suppose you have this individual here. Okay, they're linked. Okay, and it has little a, big A, okay, big B, little b. Now, just to keep in mind, this is sometimes called in trans or in repulsion, okay, as opposed to if big A were over here, and you would say it would be in cis or coupling, and that is a sort of definition based on the respective association of the wild type alleles. Okay, here the mutant alleles, mutant recessives, would be in repulsion with one another or in trans with one another. Okay, so this individual has this particular gene, genotype on chromosomes, and we're going to call this 10 centimorgans apart. Okay, and then there's another chromosome, and this individual is going to be big C, little c, like that. Okay. Now, this individual is going to be crossed with an individual that is of this kind. Okay, so little a, little a, little b, okay, and little c. Okay, and we want to, in this particular case, ask what's the probability that from this particular cross we yield an individual in F1, okay, that is homozygous for all the mutant recessive alleles. So little a, little b, little c, little c. Okay. So what is that? Well, first we just have to, well, it's easy in this particular case. I've set it up this way. This individual here only transmits little a, little b, and then little c alleles, of course. So we can just sort of designate this individual like this. The gametes that this individual transmits in that particular fashion. Now we have to think about the gametes that this individual transmits. Now, what's the probability that this individual, forget about the C allele for now, okay, that this individual transmits a recombinant individual that's little a, little b, okay? Well, we know that 10% of the time they're recombinant, okay? And what I'm going to do is sort of outline a sort of a scheme here to sort of identify all the particular gametes that are presented. So this individual produces a little a, big B, or a big A, little b, okay? And those are the parentals, okay? You can do a P for that, okay? And then the recombinants are little a, little b, and big A, big B, of course. Okay, and then those are, so the recombinants, okay? Now, what percentage of each of these classes? Well, you know, these are going to be 90% about expected, and these are going to be about expected 10% of the time, okay? But we're not just simply asking what's the probability of recombination, we're actually asking the probability of recombination of a certain kind. So this kind, so actually half of them, okay? Half of the recombinants are little a, little b, the other half are going to be big A, big B. So the probability that we inherit this particular chromosome set, little a, little b, probability is equal to 0.05. Okay. Percentages are kind of tricky to multiply. It's probably better to say 0.9 and 0.1 as frequencies. Okay. So 0.05, and then what's the chance that this one comes as little c? Well, it's 50-50, right? So the probability that this one is little c here is equal to, well, 0.5. Now, what the relationship of these is independent of the inheritance of these. So these are independent probabilities. So the probability that you inherit this entire set, okay, is half of that. So it's equal to 0.05 times 0.5, which is equal to 0.025. Okay, so half of your individuals will have this particular genotype in that particular case.